Perfect. Just started. So Ray, you've just said that you just identified a specialized cell, which is a root hair cell. Okay, so large surface area to volume ratio. You're right, to help draw up the water. Okay. What to absorb water? What else do we need to know? So we're thinking again. You remember our three things of an efficient transport system. So one's a large surface area to volume ratio. What else do we have, Ria? Can you remember any of the other two?
I think it's to maintain a concentration gradient.
Yeah, perfect. So it does a root hair cell, but you can't really see that by the structure of the cell, can you? That's more about it being taken away in the vessels. So remember we learned about the xylem and the phloem and all that. Do you remember what the last one is? So maintenance concentration gradient, large surface area to volume ratio. Do you remember the other one? Or Salma, do you remember? You're not sure, Salma? Short diffusion distance, perfect. So that's your third one. So maintenance of a concentration gradient, high surface area to volume ratio, short diffusion distance. So our root hair cell, really, really thin cells, okay? Maybe in certain places only one cell thick, like an alveoli, okay? So water can easily move from the soil into the root hair cell, okay? So another specialized part of our root hair cell, as you can see in it, it's got this thing called a vacuole. Either of you know what a vacuole contains? Yeah, perfect. So you both got it right. So cell sap. So perfect. So an important thing about cell sap is it's quite, well, it's a hypertonic solution. That's what we'll call it. So it's quite sugary, okay? That means the water potential is quite low in it. So that really helps the root hair cell because it means that the water potential in a root hair cell is always really low. So water is always want to move in via osmosis from the soil into the root hair cell to try and increase that. Okay, so that's why vacuole is really important. All right. Now another thing a root hair cell has is it has loads of mitochondria. Why do you think it might need loads of mitochondria? Anyone know? Salma, so you put energy, yeah. So it needs lots of energy. So it actively transports lots of minerals from the soil and things like that. Okay, so we need our active transport. All right, perfect. So thanks, Rita, for that. So that's our root hair cell. So Salma, I can see you've sent in the chat lots of different cells. Let me just go back up and see which one you put first. So a red blood cell, Salma. So do you know any, you can tell me in the chat, do you know any ways that it's adapted to its function, a red blood cell? Or really, you can write in the chat as well if you know any. Perfect, yeah. So Samra, you've both put two separate things, which is great. So Samra, you put no nucleus. Okay, so it's specialized, it has no nucleus. Okay, that means that there's loads and loads of space inside the cell. We don't need a nucleus. Okay, we want to carry oxygen, don't we, around the body? That's the whole point of a red blood cell. Okay, so by having no nucleus means there's more space for our hemoglobin to be in there, which we may learn about next lesson or today if we get time. Okay, more space for hemoglobin, more space for oxygen. Perfect. And then Ria, you've put a biconcave shape. Perfect. Do either of you know what advantage a biconcave shape gives to our red blood cell? Do you know what it does? So Samuel, so you put flexibility. So the flexibility is an adaptation of red blood cell, but it's not due to the biconcave shape. So we'll come back to that. So Ria, perfect. So the biconcave shape increases the surface area. Again, it all comes back to that surface area to volume ratio. Great. So Sammy, you've put about flexibility here. Okay. So it needs to be quite flexible, this red blood cell, because remember when it goes through arteries, you know, they've got quite a big lumen, goes through arterial, but when it goes through a capillary, that's really, really small. It needs to squeeze through that tiny hole. Okay. So it needs to be quite flexible. So it's got like an elastic membrane or elastic fibers in it that helps it to become quite flexible. It can change shape a little bit, so it can squeeze through really narrow capillaries. Okay. And one more advantage that it has, or specialized cell, is that it's got hemoglobin in it. So something I've already mentioned, and that's what helps carry our oxygen. All right, perfect. Right, so Ria, have you got another cell that you can identify from here?
The sperm cell.
Sperm cell, perfect. And do you know any adaptation of a sperm cell?
It got the flagella for movement.
Yeah, perfect. So that helps it move across like the fallopian tubes and the uterus and all that. Yeah, perfect. Okay. And another specialty it has is it just contains half the number of chromosomes that's needed, okay, in a embryo. Okay, perfect. So Salma, you've put here lots of mitochondria. Perfect, because it's going to need to, you know, power that flagella somehow. Okay, perfect. Anyone know any other specialization it has or adaptation?
Is the head made of protein?
Yeah, perfect. Well, proteins, kind of. So do you know what specifically it has in the head that helps it with its function?
No.
So it's got digestive enzymes. Okay. So yeah, so enzymes are a type of protein too. You're right, Ria. Oh yeah, Salma, you've put enzymes here as well. Perfect. So it's got digestive enzymes which can help break down that outer layer of the egg when it comes to fertilization. Okay. Perfect. Okay. And I think that's all the adaptations. All right. Salma, have you got another? Well, let's go back up to what you put, Salma, because you sent me a lot before. So you put a guard cell, Salma. So our guard cell is this green one on here, isn't it? So where do we find guard cells, Salma? So it's this one here. And if you're not sure, just put a question mark, that's fine. Yeah, so Ria, so you're right. So you put it's found in plants, but do you know, does anyone, and either of you know specifically where it's found? Yeah, so you both put the leaves, but a little bit more specific. I can't say that word, specific. Perfect, Ria. So you put stomata. That's great. So it's around the stomata, and it can open and close the stomata depending on certain conditions. So remember, we learned about stomata a while ago. Is the stomata is where water vapor is lost from, isn't it? So if we've got a really dry day, we probably want to close our stomata to stop water being lost. All right. So our guard cells can either become really turgid, so they can become filled with water. Okay. And that means that they open the stomata. So if a plant's well hydrated, the guard cells are going to be filled with water, turgid. So remember, a plant cell doesn't burst, does it? Because it's got that cell wall. Okay, they just become really turgid, really, really full. Think of it like a balloon that's near to bursting, but it doesn't burst. Okay. And when the plant's a bit dehydrated, not got much water, they become more flaccid, so not turgid, and that means that the stomata closes. Okay. So some adaptations that a guard cell has is that the cell wall is really quite thick. Okay, so this prevents any bursting, because remember, it can become really, really turgid. Okay, and it allows it to bend slightly to open and close that stomata. Okay. Okay, great. So that's about the guard cells. Right, Ria, have we got another one on here, what we can see? Can we identify another cell? So cilia, yeah, so this one here. So cilia is what's on the top, Ria. So the cell is actually called a ciliated cell, okay? Do either of you know where we would find a ciliated cell? Perfect, Ria. So in the trachea, remember, I think we learned about that, didn't we, when we did exchange surfaces a couple of lessons ago? Okay, perfect. So the whole function, isn't it? Do you remember? It's like the Mexican wave, we kind of said, that it's waft up debris and mucus and things like that out of the lungs to prevent any infection or anything like that. So how they're adapted is they've got cilia on the top, okay? So that's the main way they're adapted. And remember, they also come with these other cells called goblet cells, and they're what secrete mucus, and then the cilia waft the mucus up. So remember, the goblet cells secrete mucus, trap all bacteria, dust, debris, that kind of thing, and then the cilia on the ciliated cell waft it up, and you either cough it up or swallow it, and the acid in your stomach kills it. Right, perfect. Right, let's have a look. Salma, what else you put? Oh, so I think we've done all of yours. Good, Salma, can you identify any of the other ones? So what have we got left? We've got this one here, this one, and this one. Okay, so Salma's just asked, is it only feature the thick cell wall with the guard cell? Yes, kind of. It does contain chloroplasts and things like that, but we don't really know, the point too much of those. Okay, not too much, but it's got quite a thick cell wall in certain places and quite a thin cell wall in other places, so it just helps it bend. Okay, because the whole point of it is to open and close that stomata. Yeah, right. So Salma's put this one. So epithelium cell. So you've put epithelium cell for this one. Okay. So there's a couple of types of epithelial cells. So this one is what we call a squamous epithelial cell. Okay. So if I say that this is what surrounds an alveoli, an epithelial cell, what do you think the adaptations of an epithelial cell are? What do you think? If you think of the adaptations of an alveoli, don't know if anyone's thinking. Right, so remember those three things that make a good diffusion path. Oh, so Samuel, so you've put one cell thick, so perfect. So an alveoli is one cell thick and it's one squamous epithelial. Oh, Zaroe's just joined, just admit her. Okay, perfect. So it is one cell thick, and it means that it's quite thin and can allow that. Hey Zaroe, do you want to switch camera on for me?
My Wi-Fi wasn't working, so it won't let me turn anything on. But okay, I can do it after the break if we have one.
Yeah, no problem. Just if you can talk, that's great, or send messages in the chat. We're just going over specialised cells, and then we're going to start on stem cells. All right, so you've not missed too much, don't worry. You'll just have to have a read up on specialised cells that are in your specification. All right, that's what we're just going over. So we're just on our last few. So we've just gone over our squamous epithelium, okay? So it's really thin. So it's just one cell that's been flattened. So it's really thin, allows that really short diffusion pathway. And think of the alveoli, oxygen goes out, carbon dioxide goes in. So it allows gases to go through really easily, okay? So Zareen, we've just got these two left. If you can see my laser pen, can you identify either of these? This one might be a little bit easier to identify, if you can.
Is one of them an animal cell? I mean, yeah, no, a plant cell, sorry.
Yeah, so this is a plant cell, okay, but it's specific type, so it's a palisade cell, okay, so you are right, yeah. So it's just, so you may have heard about them in GCSEs before. All right, so we'll just do our adaptations. Okay, so you probably, does anyone know any adaptations of a palisade cell? You might not. Okay, so no problem, we'll go through it. So the whole point of a palisade cell is involved in photosynthesis, okay? So it's where photosynthesis can take place, all right? So it contains chloroplasts, these little ones. It's got mitochondria. It's got a nucleus. It's got a vacuole, as we've talked about it. Okay. So, sorry, just got a few questions here. So let's just go through them. So Salma put is the only feature of the short diffusion pathway one cell thick in a squamous cell. So it's permeable to gases, squamous cell, and it's just flattened really thin. Yeah. So the whole point of a squamous or epithelium is it's quite thin. Okay. So allows exchange. It's a quite good exchange surface. All right. So they're the ones. All right. So your palisade cell, yeah. So they're really quite tall and thin. All right. So they've got lots of chloroplasts in. So this is their adaptations. Okay. Lots of chloroplasts. So this means it can absorb maximum amount of light that we need for photosynthesis. Okay. And they're also tall and very thin. All right. So this means we can pack lots and lots together next to each other. But they're still all exposed at the top to allow light to be absorbed. All right, so that's our palisade cells. This is where photosynthesis takes place. All right, so I'll do our last one. I'll let you all get out of it, this last one at the bottom here. So we're looking at this one here. So this is actually a white blood cell. All right, so we've done our adaptations of our red blood cells, but we've done our white blood cells. Okay. And the one specifically they want to know about in specification, so this is the OCR specification, but AQA, you have to know about it as well, and I'm sure other exam boards, is about a neutrophil. Okay, so it's just one type of white blood cell. We've got quite a few. Okay, so the whole point of a neutrophil is to destroy pathogens by phagocytosis, which is kind of like eating it. All right. And when we do all about the immune system and stuff, we'll learn a bit more about neutrophils. Okay, so one adaptation is, again, like red blood cells, they're very flexible, all right? So they can squeeze through little holes in the capillary wall and can squeeze through capillaries as well. Okay, so they need to be quite flexible. Okay, and another adaptation is that they contain, a lot of them contain digestive enzymes, what we call lysosomes. Okay, and this helps them when they engulf that pathogen or bacteria, it helps them digest it and break it down. Okay, so that's the first part of the immune system. It's involved with neutrophils. They engulf the pathogen and break it down with the digestive enzymes they've got in them. Okay, so perfect. So a big thing is that it's flexible, and you can see here, this is what the nucleus looks like. All right, in a neutrophil, and that's what we call a bilobe nucleus. It just means it has multiple lobes. So normally a nucleus, as we've learned about, if you look at this palisade cell, it's just, this is what you've probably seen before. But a neutrophil has this bilobe, and that helps it be flexible as well. You can imagine it squeezing through a capillary, can't you? Okay. So perfect. Well done, guys. You ordered really well. So they are, how many are of them? 1, 2, 3, 4, 5, 6, 7, 8. So they're the 8 cells in specification that I mentioned. Okay. So that's possibly what could come up in exam. They might ask you about different specialized features of each of those cells. So make sure you've got a couple of points for each, and I think that'll be really useful. Okay, perfect. So as I said, this is your specification point I always write at the top here. Okay, so we need to know about the features and differentiation of stem cells. Okay, so before we move on, does anyone know what a stem cell is, or a definition? It doesn't have to be the official definition, but does anyone know what it is? And you can write in the chat if you want to, guys. Yeah, that's good. So Salma, so you've put a cell before it specializes, or it can specialize. Perfect. Right. Great. So perfect. So it is. Okay. Perfect. Zero in, so it can differentiate. Okay. Into a desired cell. Perfect. So there are two main key words here: differentiate and specialize. Okay. So a stem cell is a cell that can differentiate and specialize into a certain type of cell to perform a specific function. Okay. So that's what we need to know. So it can divide an unlimited number of times. Okay. So we've talked about mitosis, haven't we? So it can undergo mitosis. Okay. And each of the cells can either stay as a stem cell or can differentiate and specialize into a certain type of cell. Okay, so I want each of you to just give me a type of cell that a stem cell could differentiate into. Everyone just give me one. So just a type of body cell that we could make from a stem cell. So Sammy, so you put skin cell here. So Zaraween, so you've put a type of stem cell. So just give me a type of body cell. There's loads and loads of different types here. You can give me multiple if you know. Okay, so two of you have put an embryo. Okay, so we'll talk about an embryo in a bit. So that's more of a type of stem cell, but if we just talk about a type of body cell, just give me one. So Salma, so you've just put skin cell, okay. So let's go through it. So I've just got a few here. So a muscle cell, fat cell, bone cell. So just obviously a very simplification of the cells. We've got nervous cell, epithelial, immune, so our white blood cells, and our sex cells, so our egg or our sperm here. So we can differentiate into, our stem cell can differentiate and specialize into all of these types of cell. So a specialized cell is a cell that can perform a specific function, okay? So a muscle cell obviously works together to contract a muscle to move bone, okay? A nervous cell relays action potentials or relays like a nervous impulse, okay? A red blood cell, that's what carries our oxygen around our body, and so on and so forth, okay? So that's why we need these stem cells to be able to differentiate into. All right, and we need to know about the specific type of stem cells that we've got, okay? So these are the specific types that we normally talk about when we talk about stem cells. So we've got our totipotent stem cell, our pluripotent stem cell, and our multipotent stem cell, okay? And each of them are stem cells, but they're all ever so slightly different, okay? And you need to know the difference between them, really, okay? So if we start with our totipotent stem cell, this is what everyone thinks all stem cells are, okay? So this one here. So this stem cell has the ability to differentiate into any type of cell in our body, okay? Any type of cell whatsoever, okay? It can also differentiate into cells that will make up the placenta, okay? So when you're developing, obviously you've got the fetus, you've got the baby, the umbilical cord, and you're attached to the placenta, okay? So a totipotent stem cell can make any type of cell that's found in an embryo and the placental cells as well, okay? So if we think this is probably our best type of stem cell, okay, because we can make absolutely anything from it. All right. So think of this as kind of like a little line. If I draw little lines on it, if I draw little arrows, and as we move further down the arrow, they differentiate into less and less types of cells, okay? So Ria's just put, do we need to know the pluripotent stem cells? Let's just have a quick look. So in your specification, it just says the features and differentiation of stem cells. Okay, so that's what it says, and then we're going to be learning about a specific type. All right, so we'll just go into it and learn them all, but I think definitely learn, if you can learn all 3 types, that's great. Okay, and it depends on different exam boards as well, because I know some of you are doing AQA and some are doing OCR as well. Okay, so totipotent can go into any type of cell in the embryo and placental cells. Pluripotent can go to any type of cells in an embryo, but it can't differentiate and specialize into placental cells. Okay, so that's the difference between totipotent and pluripotent. So like I said, as we go down the arrow, we lose a type of cell we can differentiate into. Okay, so again, totipotent and pluripotent, we'll only really find them in embryos. Okay, so that's the only place we'll really find them. All right, so then we move down the arrow again and we move into multipotent cells. Okay, so these can still differentiate into many types of cells. Okay, but it's only a very specific group of cells. Okay, and the big example, so I'll answer questions in a second when we go. Okay, so the big example, okay, that we always use is we find multipotent stem cells in someone's bone marrow, so inside the bones, and it can differentiate into any type of blood cell. Okay, and we'll learn about that specifically. So Salma, so you've just asked, sorry, what's the difference between, so is that difference between totipotent and pluripotent, I'm guessing? So the difference between totipotent and pluripotent is totipotent and pluripotent can differentiate into any type of cell within an embryo, so any type of body cell, but totipotent can also differentiate into placental cells, so cells we find in the placenta, but pluripotent can't differentiate into placental cells. So that's the difference between those two. Okay, so small difference, but it's important. Okay, so totipotent, like top rank, can differentiate into anything at all. Pluripotent, anything in the body, so embryonic cells, so not placental. And multipotent, only a very specific few cells it can differentiate into. Okay, so totipotent we find in a really early embryo, so when an egg and a sperm fertilization in a very specific point. Okay, so it's only been a couple of days that the embryo has been made. Then once the placenta starts to differentiate and specialize off, then we get pluripotent stem cells. And then in our bodies now, so in all of yours, you'll have multipotent stem cells. Okay, great. So as we go down, we can differentiate into less and less types of cells. Okay. So the example you guys need to know is about erythrocytes. So that's a red blood cell, and neutrophils, which is a white blood cell, which we've just learned about, haven't we? Derived from stem cells in the bone marrow. Okay, so let's have a quick look. So these are, don't get too worried about everything. Oh, sorry, let me just delete those arrows. These are all the different types of blood cell that our multipotent stem cell can differentiate into. So let's have a look at our key ones here. Okay, so a monocyte, lymphocyte, neutrophil, eosinophil, basophil, and macrophage, they're all white blood cells. Okay, so they're all involved in your immune system. So the only one you guys really need to know so far is the neutrophil that we just talked about. So just concentrate on that. I would ignore the rest so far. Okay. And then we've got our erythrocyte, which is another name for our red blood cell. So that's what carries our oxygen around our body and contains our hemoglobin. And then we've got our platelets. Okay. So platelets are another form of blood cell. And that's what helps, you know, if you cut yourself or get a scab, that's what helps clot our blood. Okay, so it's important. All right, so we talked before, if we just concentrate on our red blood cell for now, we talked before how about our red blood cell doesn't have a nucleus, okay? It's biconcave and it's really flexible. Remember, those are our three main adaptations to what we need. Okay, so we need to think how we get from our stem cell, which is, you know, let's just say it looks like a regular cell. So let's just draw one up here. So we've got that. We've obviously got a nucleus. Let's just draw it with our nucleus. How we get from that type of cell to our red blood cell, okay? So basically, we need to get rid of our nucleus, don't we, to form from our multipotent stem cell into our erythrocyte, our red blood cell, okay? So, like I said, this cell's multipotent. It can form any type of these blood cells that we can see here, okay? So what it starts to do is it starts to differentiate and it starts to specialize into an erythrocyte, okay? So the adaptations we need to do is we need to get hemoglobin into the cell, okay, so we can carry out oxygen. We need to get rid of our nucleus as well, okay? So you guys don't need to know too much detail about the process, but you just need to know it starts to differentiate, okay? So it starts to change. So now it's made small changes, so now it can only form our red blood cell. It starts to get a build-up of hemoglobin within the cell, okay? So that's important, that's what we need to carry oxygen. And then it just gets rid of its nucleus, so it gets rid of its DNA. So just think of it, it throws it out the window like if you're in a car, okay? And then it starts to specialize and differentiate even more, gets that biconcave shape, becomes really elastic to form our erythrocyte, okay? So it's really easy if you remember your three adaptations, that it's got no nucleus, it's got hemoglobin, and it's elastic. And you just say that in there. So it needs to build up its hemoglobin, it needs to get rid of its nucleus, and it just needs to change shape slightly to become that biconcave shape. Okay, perfect. Great. So that's how we get how we make our erythrocyte from our stem cell. Okay. So now we need to concentrate on making our neutrophil, okay? So like I said, we'll just concentrate on neutrophil as our main white blood cell. Like I said, there's lots and lots of different types, all right? And they've each got slightly different functions. So again, we're starting with our multipotent stem cell, okay? So we're starting with the exact same cell that we form our erythrocyte from, okay? So what we need to get our neutrophil, remember its adaptations, is it's very flexible again. It's got a bilobe nucleus, so that enables it to squeeze through, okay? Because if you think if the nucleus was like a solid circle, like in most cells, it wouldn't be able to squeeze as much, but because it's got different bilobe parts, it can squeeze through one part by one, can't it, through those small capillaries, okay? And we also need to get those digestive enzymes in it. Remember those lysosomes? Because when we attack pathogens and things, it can break it down, okay? So again, so what we need to do here is we need to get a buildup of lysosomes within our cell, okay? So we need to make lots of lysosomes. So we need to start to differentiate and specialize again, okay? And then we need to kind of make our nucleus bilobed, okay? So we need to kind of squeeze off little areas, make, you know, and make little, if you think of it, they look like, so our nucleus is going to end up kind of looking like that, okay? So bilobed. So we need to squeeze in this little bit here and squeeze in this little bit here. Again, you don't need to know the details. You just need to know how it's specialized, this cell, okay? And that will help you. All right, perfect. Great, so they're just the two ones that we kind of need to know about, all right? As you can see, that's your specification point a bit. So the production of erythrocytes and neutrophils derived from stem cells in the bone marrow, okay? So important points here, these are our multipotent stem cells. They can form red blood cells, all these types of white blood cells, and our platelets, okay? And it's found in the bone marrow. So that's kind of inside the bone. And you may need to describe how the erythrocyte becomes specialised, or how the neutrophil becomes specialised. So you just need to describe different ways that they're specialised, okay? So maybe remember 3 for each. I think that'd be really useful. Okay, perfect. So just conscious of the time, and we want to, so we were going to split the lesson in half. All right, so perfect. Okay, so you also need to know about plant cells, stem cells. Okay, so we did plants a few weeks ago, didn't we? I remember the 2 main vessels in plants, a xylem, okay, and that's what carries the water, basically just the water, and the phloem, that's what carries the sugar, so the sap, okay? So we need to form xylem and phloem from our stem cells, okay? So within a plant, our stem cells, if I get my little laser up again, our stem cells are found in this bit called the meristem, okay? So you can see it in between the xylem and the phloem. You can see that in this. So remember the stem, this is what this cross section looks like, okay? And then the root here. So remember, xylem's always in the middle, and in the root it makes that X for xylem, okay? So the cambium, it's called, the meristem, is where we get these undifferentiated, unspecialized cells, okay, that can form new cells. So kind of like a multipotent stem cell, they can either form a xylem or the phloem, okay? So they're found in the stem, they're found in lots of places, they're found in shoots and roots as well, okay? Sorry, shoots as well, and then they're found in the root here, okay? So in the stem, important to remember, it's in between the xylem and the phloem. And in the root, again, it's right on the edge of that xylem. So again, in between the xylem and the phloem. Okay. So we need to know what causes them to differentiate. Can anyone think what may cause them to differentiate? How might the plant communicate to this cambium or meristem that we need more xylem cells or we need more phloem cells? Can anyone think? And if anyone's stuck, think of in your body, how does your body communicate to other parts of your body? Riya, perfect. So you put neurons here. So that's one communication. So that's your nervous system, isn't it? Well done, Riya. So your plants, I'm not sure, they may have a nervous system, but I'm not aware of it. So it's not your nervous system in this example. So there's another way your body can communicate with others. So you might not get it, guys. So it's with hormones, okay? So hormones is a chemical messenger, okay? So we can send different hormones across our body, and they turn on things, turn off things, and communicate in different ways, can communicate with different cells, okay? So that's what's involved at this stage, okay? So to make our meristem into either xylem cells or phloem cells, hormones are used, okay? So that's what the plant uses. Don't need to know any more details than that, don't need to know the specific hormones, okay? So we can do that. So quickly before we move on, what specialized features a xylem has and what specialized features as a phloem has. Okay, can anyone remember any key features of a xylem cell? We learned about them quite a few weeks ago now, but can anyone remember any? You can shout it out if you want to, if anyone knows. Perfect, Irene. So they lose their end cell walls. So remember, you need to make a really long column for water to go through. Remember, we've got that really thin stream of water, their adhesion, cohesion, okay? So they lose their end cell walls. Perfect, Zoe. I'll give the rest, okay? Oh, perfect, Ria. Lignin cell wall. Lignin in the cell wall, as well as cellulose, makes it really, really strong, okay? So it provides structure to the plant, all right? And then the last adaptation of xylem is they lose basically all their organelles, okay? And it makes them hollow, okay? So that's how we adapt with our xylem. So if you get a question, how does a cell, stem cell, or how is a xylem cell created from a stem cell? You can talk that plants have stem cells called the cambium or the meristem. Hormones communicate with the cells to form xylem cells. The cells then lose their end cell wall, they lose all their cellular content, and they get lignin in their cell wall to provide structural support, okay? Easy, that'll be like a 4-mark answer there, all right? Perfect. So if we need to make phloem, then what's the difference? What's some adaptations of the phloem? But no one can remember any. Perfect, Ria. So has sieve end plates. So they're end cell walls. So they don't lose them completely, but they're called sieve end plates. So think of it, it's just like a sieve, all right? Have little holes in that allow things to go through, okay? So remember the difference in xylem and phloem. Xylem doesn't have lignin in it, it just has your typical cellulose, okay? And another adaptation of the phloem was it has companion cells next to it, doesn't it? So that's kind of not the phloem cells themselves, okay, but the phloem tube has companion cells next to it, okay? Perfect. Yeah, Maria, you're right. So the sieve end plates allow two-way movement. So remember, the phloem wants to take something from the source to the sink, okay? So the source is going to be the leaf, so where photosynthesis takes place. We need to take the sugar produced from that to somewhere else. So our sink could be our roots, or it could be a piece of fruit at the top of the plant, okay? So we need to allow movement up the phloem and down the phloem, okay? Because we don't know where it goes, where the xylem just goes up from the roots to the leaves. Perfect. Well done, guys. So remember those really well. So the last part before we'll have a quick break, okay, that you need to know about is the potential uses of stem cells in research and medicine. Okay, so what type of stem cell do you think is best to use in research or medicine? So remember, we've got our pluripotent, totipotent, multipotent. What type of stem cell do you think is best? Does Ria jump say it out loud? You got the answer right?
Totipotent.
Yeah, totipotent. Perfect. Okay. So totipotent, because as we've talked about, they can differentiate into any type of cell possible. Okay. So that's what we want in our research or medicine, is we want to make any type of cell. So why would we pick a multipotent cell that can only differentiate into, you know, either a red blood cell, white blood cell, platelet, or another type of multipotent cell? We want to pick that one. So thinking about where we get totipotent cells from, what do you think maybe the disadvantages or the problems with using totipotent cells in research? Can anyone think of any problems? So perfect. So Salma, so you've put ethical issues. Okay, perfect. So a few of you put it's unethical. Great. So remember, we find our totipotent and pluripotent cells from embryos, okay? And it could be unethical, and there's lots of ethical dilemmas about it, all right? One is that those embryos could potentially be implanted, you know, in someone's uterus and become a human being, okay? So are we taking away someone's life? That's a thing. Or would that embryo never develop anyway? Would someone just get rid of that embryo? Okay. So it's weighing up the pros and the cons, and you might get a question on this, you know, evaluate the use of stem cells in medicine, okay, or the use of totipotent cells, okay, or the advantages and disadvantages of using totipotent cells, okay. So that's kind of unethical, isn't it, to maybe take away from an embryo. It's obviously, some people agree with it, some people don't. That's why it's an ethical issue, okay. So basically we need to use multipotent cells, really, because they come from an adult and they can consent to giving their stem cells, okay, and we can make more stem cells within our body. It's not taken away from embryos, okay. So has anyone heard of a treatment where we might use multipotent stem cells from a human? Does anyone know a treatment? Perfect, Ria. Just see if anyone else knows as well. Great, so Ria's put a bone marrow transplant. Perfect, Ria, yeah. So you guys might have heard you can donate your bone marrow to someone else, all right? So the example that we always use is leukemia. So that's a type of blood cancer, okay? And that happens when someone's stem cells within their own body, when they differentiate and specialize, they produce cancerous blood cells, okay? So you can have different types of leukemia. Sometimes it can just be a specific type of white blood cell. You know, remember there was about 5 there, wasn't there, that we did? It can be all of them. It can just be a couple, okay? So their stem cells are differentiating and specializing into cancer cells, okay? And that's how they get cancer of the blood. All right, so a treatment to try and eliminate that cancer from their body is to give them chemotherapy, which I'm sure you've all heard of, to kill all of their stem cells, their multipotent stem cells that go on to create blood cells, okay? That's part of the treatment. So we want to try and kill all of them to stop them producing cancerous cells. But obviously they can't go on living like that because they can't not create any cells in the body. So what we do, as what Ria's put, is we give someone a bone marrow transplant, and bone marrow contains multipotent stem cells, okay? So let's say that I had leukemia, so we're getting rid of all of my stem cells, and Salma is giving me a bone marrow transplant, okay? So I'm going to get Salma's non-cancerous multipotent stem cells, and then they can start to divide and differentiate and specialize into white blood cells, red blood cells, platelets, okay? And then I can just use Salma's stem cells for the rest of my life and do like that, okay? Does anyone know any problems with a bone marrow transplant? What's the biggest problem? Yeah, perfect. So a few people have put it. So it's rejection, isn't it? And infection. Perfect. Yeah. So there's a few actually. Yeah. So the biggest one I can think of is rejections. So me and Salma aren't the same person at all. Okay. I assume we're not related in any way. So we're not going to have our little antigens on the surface of our cells are not going to be compatible. So when I get Salma's stem cells in my body, they're going to look like an invading cell. They're going to look like a foreign cell, okay? My body is going to attack them, okay? And they have to give someone anti-rejection medications, okay, to stop my body attacking them, all right? And that can cause lots of different side effects. As Irene, you're right, infections, anything to do with like an operation or anything like that can cause infections or bleeding risk, or it can just not work, okay? Or my body can start producing the cancerous stem cells again, okay? So there's lots and lots of different things, all right? So that's one example, and it's the main example that you'll probably get in the exam. But you know, there's lots and lots of different opportunities that we could do with stem cells. If you think if someone's got, you know, been paralyzed, they've damaged a spinal cord, maybe we can make spinal cells and inject them into someone's spinal cord and they become not paralyzed, be able to walk again. If someone's, you know, got diabetes and their pancreas isn't producing insulin, we can give them pancreatic cells and can start producing insulin. So Salma just asked a question, would you have to have both chemo and bone marrow transplant, or is one enough? No, you'd have to have both, because the chemo would get rid of my cancerous cells and the bone marrow transplant would give me healthy cells, okay? So I can't produce healthy cells by myself. I'm only producing cancerous cells, because that's what all my multipotent stem cells are. They're all cancerous, okay? But if I get your stem cells, Salma, that aren't cancerous, if I get rid of all of mine first, eliminate them, and then get the bone marrow transplant, then I can produce healthy cells, okay? So perfect. So there's, you know, lots and lots of different uses of stem cells within science and within medicine, okay? Perfect. Right, so that's just our last one as well. Oh, one thing, I'm just looking at specification, the OCR one. So they've said about, it says to include the repair of damaged tissues. Okay, so think damaged tissues could be, like I said, the spine, or if someone's had a heart attack, that's damaged part of the heart, you can give them stem cells that can differentiate into cardiac muscle. Okay, they've also put the treatment of neurological conditions such as Alzheimer's and Parkinson's. Okay, so Alzheimer's is a form of dementia, isn't it? Okay, and maybe we can give brain cells to rejuvenate and regenerate someone's brain cells. Okay, and then Parkinson's, okay, again, another condition within your brain. So we can give these stem cells to maybe create new brain cells, and so they can produce all these hormones and these messengers, okay, that they've stopped producing and now they've got Alzheimer's or they've got Parkinson's or something like that. Okay, so lots and lots of different uses. All right, perfect. Well done, guys. So I think that's the end of that PowerPoint there. So what we'll do, should we have a 3-minute break to lead us up to 8 o'clock, and then we'll do half an hour on the structure of the heart and carrying on for the next lesson? Is that all right, guys? Want to go and grab a drink? Someone just want a message, or just speak out loud? Yeah, perfect. All right. So if we come back, if we come back at 1 minute past, all right, and be ready to learn about the heart, okay? everyone could just send a message in the chat when you're back, so we know when to carry on. Perfect, thanks, Sarah. Thanks, Zoe. Just wait for Ria then. Great, so we'll just do half an hour of a bit on the heart, and then next lesson we'll get some exam questions for that stem cell bit, and we'll get some more exam questions on the heart as well. And let's see if we can try and finish off our transporting animals. We'll see if we can do it. So we'll get started because don't want to wait too much time. Okay. So perfect. So we did quite a bit last lesson. If we just go through these slides. So we did why we need a transport system. Okay. We did about our single and double circulatory systems. Okay. So remember, single goes through the heart once in one complete cycle, double goes through the heart twice. All right. Oh, hey, Ria. And we did about our open and closed as well. So we did our closed was our humans or our fish, and our open was our insects. Okay. And remember, insects could have an open system because they don't rely on it for their oxygen transport. Okay. And then we did about blood vessels, our arteries, our arterioles, capillaries, veins, and venules. Okay. So we're going to be doing a little bit more about arteries and veins. Okay. And then we also did about the structure of the heart as well, which we can go over again. Okay, if we need to. So we've still got quite a bit to do on this topic. So let's just see how much we get through in the half an hour. Okay, so let me just turn it on to PowerPoint. Okay, great. So we used these last time about the structure in the artery and the vein. So the main thing about the artery was it had, a high amount of muscle. Okay, and that allowed it to constrict the lumen if it needed to change the pressure. Okay, it also had elastic fibers in its wall, and that allows the stretch and recoil because it's right near where the heart pumps. Okay, so when the heart pumps, it needs to stretch, and when the heart relaxes, it needs to recoil back. Okay, so that was important. Okay, I remember the arteriole again was very similar, had less elastic fibers, but it could also had high amount of muscle because it also constricts the lumen. It can control where the blood goes to. All right. Then we learned about the capillaries, that they're 1 cell thick, okay, to allow that exchange of substances across, and they're really, really narrow as well. Okay. And then we did about veins, is they've hardly got any elastic fiber and they've hardly got any muscle. Okay, so they move, they can be squashed because they've hardly got any muscle. Okay, so they move the blood up by compression. So if I think it was Sarah, I said if she flexes her arms, okay, last lesson, then the blood in your vein is going to shoot up because the vein's going to be compressed by the muscle. Okay, and then when you relax your muscle, it can't go back down because of the valves in your vein. Okay, so we learned a lot about valves last lesson, and we learned about them in the heart as well, didn't we? That's just a quick overview. So you just need to know a little bit more about the structure of an artery and a vein. Okay, so I've got this quite good image off Google. All right, and you may have to identify if a certain vessel is an artery or a vein. So what you're looking at is, remember that word, a relatively small or a relatively large lumen. Okay, and that just means in comparison. All right. So an artery has a relatively small lumen in comparison to the whole of the vessel. Okay, so you can see it's got a quite thick wall of the artery. Okay, it's much smaller, and normally they're a bit more circular arteries. Okay, a bit easier to see. Whereas your vein, you can see it's got a relatively larger lumen. Okay, much thinner wall. Okay, and you can see it's a bit of a wider, more semicircle shape, isn't it? Okay, so that's normally how you identify an artery or a vein, and normally the differences between them. just takes a bit of practice to have a look, but they're the main things. Okay. So a little bit more about the structure of the arteries and veins. Okay. So we learned that they've got muscular layer, and the arteries have the elastic fibers. Okay. So just a little bit more. So you just need to know that there's 3 layers. Okay. So the artery wall's got 3 layers. So, you know, it's quite easy to learn because they're quite logical. Okay. So you've got the tunica externa. So quite logical. You've got the tunica externa is the outside. Okay. You've got the tunica media. Okay. So that's your thick muscular layer. Okay. So that enables it to constrict and narrow to increase the pressure. Okay. And then the very inside, you've got your tunica intima. Okay. So think of it as tunica externa, EX, exit, outside. Okay. Tunica media, middle for M, and tunica intima, I for inside. Okay. So your tunica intima's got, you'll have your endothelial cells, it's got elastic fibers and things like that. Okay, so remember your endothelial cells is only 1 cell thick. Okay, so they're your main 3 layers, and that's what you've got in your vein as well. Okay, so you've got the tunica, it's called they've named it something slightly different, but it's also known as the externa, which they put here. So you've got your tunica externa, tunica media, and the tunica intima. Okay, so they're the 3 layers. So you can see that your tunica media in your arteries is much, much thicker than your tunica media in your veins. Okay, and that's just because your arteries are right near your heart, as I've said, and they have to withstand that pressure from when your heart pumps and when it relaxes. Okay, so when it pumps, it's under high pressure. When it relaxes, it's under lower pressure. Okay, and that's how it stretches and recoils. All right, with the elastic fibers in the layer, but your veins are, you know, very far away from your heart. All right, and they take blood back to your heart, don't they? Okay, perfect. So they're the main things I just wanted to add in when we need to do a little bit more detail. So tunica externa, exit, outside, media, middle, intima, inside. Okay, so it's kind of easy to remember. And your tunica media is the kind of the difference between your arteries and vein. Much thicker in your arteries, much thinner in your veins. Okay, they're the main bits I wanted to put. Right, we'll go back to tissue fluid in a second. What we kind of ended on last week was we were doing the structure of the heart. Okay, so let's just quickly summarize very, very quickly, so don't spend too long doing it. So remember, we go in, if I draw our blood with this, so we go in our vena cava. So remember, there's 2, the superior and inferior, but incorrectly labeled on here. We go into our right atrium. Okay, so our right side of our heart. We go through our AV valve into our right ventricle. We go up through our semilunar valve into our pulmonary artery. Okay, and then we go to the lungs. We come back here through our pulmonary veins into our left atrium, through our AV valve into our left ventricle, up here through our semilunar valve into our aorta. And remember, the left side of the heart is your pulmonary vein and aorta, and it's PVA glue. So remember, PVA, that's how you remember which sides of the blood vessels are on either side. Okay, so we learned about that last lesson, and we just need to finish off that bit about the external structure of the heart. Okay, so it's a little bit blurry, this, apologies guys, it's the biggest I could get it. Okay, so we've done this part, we've done our internal structure. So the external structure of the heart is quite similar. So you can see you've got your superior vena cava and your inferior, so it's correctly labelled on this one. So remember, your inferior comes from anywhere that's below your heart. Remember, vena cava is bringing blood back to your heart from all over your body, and your superior is anything above your heart. Okay, you can also see aorta, so that's an artery, so it's the biggest artery in your body, and these are just all the branches, don't worry about that. And then you've got your pulmonary artery here. okay, and your pulmonary vein. So the important thing to remember about the external structure of the heart is you've got these called your coronary arteries, okay, and these are what are on the surface of your heart. Can anyone think of what's the point of having these coronary arteries on the surface of the heart? Why do we need these? If you think, what's the point of having arteries going towards a tissue? What's the point of our blood? What are we transporting in our blood that's important for us? Good, guys. It's the main thing we need to be able to transport. What can we not live without if we don't transport it in our blood? Yeah, perfect, Irene. Oxygen. Okay, so remember, it's carried in our red blood cells within our hemoglobin. Okay, so we need to carry oxygen. So our heart needs oxygen as well. Even though it's pumping this blood around our body, it's not actually oxygenating, you know, the outside of our hearts. Okay, so these cardiac cells. So we need arteries and veins on the surface of our heart to oxygenate these cardiac cells. Okay, so these are what we call our coronary arteries. Okay, so these are what oxygenate our cardiac cells. So when you think about in a heart attack, okay, these arteries are what get blocked a lot of the time. Okay, and that's what causes the pain and the breathlessness and things like that, because we're not oxygenating part of our heart. Okay, and it means it can't function very well, and then that affects your whole body. Okay, so that's just basically what you need to know about the structure, the external structure of your heart. Okay, so even though our heart is pumping around deoxygenated blood and oxygenated blood, it still needs to be oxygenated itself, the cardiac cells. Okay, and that's what these are for. Perfect. Right, well done. So one key thing we need to know about is how we create, that's in this topic, is how we create tissue fluid. Now this diagram looks a little bit complicated, so don't worry about it too much. Okay, so let's draw a little bit more of a simplified one. All right, so what we're doing here is we've got an artery. If you think, we start with an artery, okay, and then we've got an arterial. So this is going to be our arterial. And remember, we go from our arterial and we go into like a capillary bed, don't we? We go into lots and lots of different capillaries, because then we have a high surface area and so on. But I'm just going to draw, for the sake of this, just one capillary. All right, and I'm going to draw it quite big, but capillaries, you know, much smaller. Okay, so I'm just going to draw one capillary. I'm just going to change to blue because then that signifies deoxygenated blood. Okay, so it's still our capillary here. And then our capillary, we go into our venule, so much bigger. Okay, so that's just simulating this one here. So this is just where our venule is at this end. And then our arterial. All right, so it kind of shows it in this diagram. Okay. So the main thing to know about the blood is when we get an arterial end, okay? Remember, it's come from the artery, it's come from the heart. It's under really high pressure. That's a really important thing to remember about the blood. When it comes from the heart, it just pumps our heart and it's under really high pressure, okay? And that's why our arteries are adapted and our arterioles are adapted. So if I change the colour here, so it's coming down here under really high pressure, okay, down our arteriole, and then it gets to our capillary bed. So remember, our capillary bed, our capillaries are really tiny, all right, can't withstand that much pressure, okay, can't do it at all. So when our blood gets down here and gets to our capillary bed, so we're just looking at one capillary here, but remember, this is happening simultaneously across lots and lots of different capillaries. It's going to be under really high pressure, okay? Not all that pressure can go into the capillary, all right? You just can't go. So what happens is a capillary is made up of, it's just one cell thick, isn't it? Okay, so if we draw some some cells, some endothelial cells. Okay, so it's just one cell thick. But between cells, we've got tiny little gaps. Okay, tiny, tiny little gaps. So what happens is when we get to the capillary, loads of the contents of the blood is forced out of those tiny little gaps. Okay, so if I just delete this bit here, so loads of content, so only very small molecules are forced out. Okay, so small molecules include gases, so oxygen that's dissolved in the blood, ions, glucose, water, okay, is going to be forced out of the blood. Okay, and it's going to go into the surrounding area. Okay, so let's say this is all what's been forced out. Okay, so there's certain things that remain inside the capillary that are too big to be forced out, and it's really important you remember these guys. Okay, so one is proteins. They're far too big to move out of the capillary. Okay, so protein, and the other one is going to be red blood cells. So they remain within the capillary. Okay, so I remember those as our two main types. So white blood cells can be forced out, glucose, ions, everything like that. Okay, so when we talk about tissue fluid, we need to talk about two main things. You can see on this diagram here. So in the green arrow, we've got hydrostatic pressure. And the blue arrow, we've got water potential. Okay, so at the arterial end, the blood's under really high pressure, and that's what we're going to call hydrostatic pressure. Okay, so at this end, we're under really high hydrostatic pressure. So that's why all these molecules are forced out. Okay, so now within here, we've got much lower hydrostatic pressure. Okay. Now, we've also forced out all the water into the surrounding tissue fluid. So the water potential of this tissue fluid on the outside is going to be high, okay? And the water potential of whatever is remaining in the capillary is going to be really low, okay? Because we've forced everything out, but we've also kept protein within our capillary, and that lowers our water potential. Okay, and if this is not making sense to you guys, we'll go back over it again. We'll do a summary. Okay, so at this moment in time, we've got low hydrostatic pressure within our capillary and low water potential, and we've got high water potential within our tissue fluid, so outside the capillary. Okay, so what happens within the tissue fluid is it surrounds organs and it surrounds cells and starts to exchange substances. Okay, so this is how we exchange things. So think of it as it's suddenly surrounding your bicep. Okay, so obviously we're going to take oxygen from the tissue fluid into your muscle. Okay, we're going to take carbon dioxide out of your muscle and put it back into the tissue fluid. We're going to take glucose from the tissue fluid into your muscle, and we're going to remove any waste products that we've got in our muscle back into the tissue fluid. Okay, so this is where the exchange of substances happen. Okay, so now we get to the end of our capillary bed. So we get to the venual end. Okay, and now we've got loads of fluid, tissue fluid. We don't want loads of tissue fluid left in our bodies. If we've got too much fluid, that's where things start to swell. Your ankles start to swell. You end up with fluid in your lungs and things like that. Okay, so it's not a good sign. So we need to try and get all that fluid back into our blood. All right. So what happens is we've got quite low water potential, remember, here within our capillary. Okay, and we've got quite high water potential within our tissue fluid. Okay, so what happens is water moves back into our blood here at this end, the venual end. It moves via osmosis from high water potential to low water potential. Okay. That movement creates pressure, okay, because we're adding things back into a vessel. So we're decreasing the volume left within the vessel, so we're increasing pressure. So the movement of water also increases the hydrostatic pressure at the venual end. Okay, so water moves back in. So we're increasing the water potential and the hydrostatic pressure. Okay, and then our blood starts moving up our venual and moves back, and we all know about that, about the compression with the muscle, and that's how it moves back up against gravity. Okay, so a lot to go over. I don't know if you guys have gone over in this in class before. Okay, just let me know in the chat if you have, or you've not. That's great, and we'll quickly go through it again. Okay, so let's get rid of all of this. So we've got blood moving into the arterial end. It's under really high pressure from the heart still. Okay, the capillary can't take that much pressure, so things are forced out of the little gaps in the capillary, so we can go that. And this is what makes up our tissue fluid. So within our tissue fluid, we've got glucose, we've got ions, we've got water, we've got oxygen, we've got CO2, all that thing. Okay, what remains in our capillary is proteins, because they're too large to pass through the gaps in the capillary, and red blood cells, again, because they're too large. Okay, so now within the capillary, not much is left. So there's quite, a low hydrostatic pressure. We've also removed all of our, majority of our water. So now we've got a low water potential. Outside the capillary, we've got high water potential. Okay, so at the end, the venual end, we get water moving via osmosis. Oops, sorry, that was meant to be blue. From high water potential to low water potential into the venual end. Okay, and the movement of water also increases the hydrostatic pressure. Okay, and then the blood carries on into the venule and then into the vein. Okay, what we'll do for next lesson is I've got a really good exam question that came up a couple of years ago on this and asked me to describe, so we'll have a really good go of that. Okay, my top tips for this is when you're talking about tissue fluid and the creation of tissue fluid and the movement, you need to always refer to hydrostatic pressure, and you need to always refer to water potential. Okay, and if you understand those two bits, you'll be able to answer the question really, really well. Okay, like I said, I'll get the really good exam question that I've got for next lesson. All right, perfect. Remember, any questions, just put in the chat for me. Okay, so let's have a little start on the cardiac cycle and do this. So I know, Salma, we did a lesson a while ago, let's just see, ages and ages ago, about the cardiac cycle. Now we've only got about 5 minutes left, so we won't have time to do it all. Okay, so we can continue on this next lesson if we need to. So I've put a little equation down at the bottom. So cardiac output equals heart rate times stroke volume. Okay, so again, you might be asked what is cardiac output. So you can do heart rate times stroke volume, or you might be asked to work it out or work out stroke volume. So stroke volume is the amount of blood that's ejected from the heart in one heartbeat. Okay, and heart rate is the number of contractions in 1 minute. Okay, so normally it's between 60 and 100, a normal adult heart rate. Okay, so you need to know about the cardiac cycle and different changes of pressure. Okay, so we've talked about is that atrium contracts and the ventricles contract. So let's do this. So let's just talk about the left side of the heart in this. Okay, and let's draw out a really rudimentary, simple heart. Okay, so that's our left atrium, left ventricle, and that's our AV valve, and then this is going up into our aorta, and this is going to be our semilunar valve. Okay, so what we need to label on here is we need to label, what do these lines mean? Okay, so this one here is going to be the pressure inside our ventricle. So I'll put ven. Okay. The blue one at the bottom, that's going to be the pressure inside our aorta. Guys, it might be good to, sorry, not aorta, atrium. It might be good to draw this out because then you can label it yourself if people got paper. But if you've not, no problem. We won't go through it all this lesson. We will do it next lesson as well. Okay, so you might just want to listen to this bit and then we can draw out next lesson. Okay. And then this red one at the top is going to be the pressure inside our aorta. Remember, we're doing the left side of the heart, but it's exactly the same in the right side of the heart. Obviously, this will be our pulmonary artery, not our aorta. Okay. So it's important to understand that when something contracts, so either the atrium or the ventricle, you'll decrease the volume inside, so you're increasing the pressure. And that's what we're measuring on this graph here. Okay, so there's three main stages to our heartbeat. Remember we said last lesson, I think it was lub dub, lub dub, lub dub. We talked about that. So lub is one thing contracting, dub is another thing contracting, and then the gap in between is the heart relaxing. Okay, so the first bit is the atria contracting. Okay, so let's just draw on here. So we've got blood inside the atria contracts and it forces it through the AV valve into the ventricle. So that's our main bit. Okay, and again, we need to talk about the pressure changes within this. So let's have a look at this first section. Okay, so we've got little lines here at the bottom. So let me draw a line to separate this first section here. Okay, so we can see the pressures here. If I get my little laser, it's much easier. Okay, so remember the atrium, the pressure inside the atria or the atrium is the blue bit, okay, and the pressure inside the ventricle is the green bit. So this is what we call atrial systole. Okay, so I'll write down all of these for next lesson. Let's just do a quick overview at the minute, so a little bit of an intro for you guys if you've not done it before. All right. So at this point, atrial systole, the atria are contracting. So we're just talking about the left side of the heart, but remember it happens simultaneously in both. So the pressure inside the atria is higher than the pressure inside the ventricle, okay? And we can see that here, all right? So that means that blood is going to go from high pressure inside the atria to low pressure inside the ventricle, okay? So at this point, atrial systole, blood is moving from the atria to the ventricle, all right? It's moving through the AV valve, or we could also call it the bicuspid valve, because this is the left side of the heart, okay? From high to low pressure. All right, perfect. So at this point, the atria finish contracting, okay? Atria finish contracting, so we've had the lub, and now we go to the dub. So now the ventricles are going to start contracting, okay? So at this point, you can see, if I get the little laser up again, that the pressure inside the ventricles starts to increase past the pressure inside the atria, okay? So that's important. So at that point, remember, blood wants to flow from high pressure to low pressure. So once the pressure inside the ventricle increases, then that pressure inside the aorta, blood's gonna want to flow from high to low pressure. So it's going to want to flow from high pressure inside the ventricle to low pressure inside the atrium. Okay, but we don't want it to do that. So remember, we've got our valve there. We've got our AV valve or our bicuspid valve, whatever we call it. So at this point, our AV valve closes to prevent that backflow of blood, okay? So here, if we look on our graph, at this point, where the pressures cross over, the AV valve closes, okay? So that's here on this graph the most important bit is to know which valves open and which valves are closed. Okay, so at that point, the AV valve closes, and that's to prevent any blood flowing backwards into the atria again. Okay. All right. So you can see there's another cross here, another cross here, and another cross here. So that's, you know, we need to talk about more about which valves we're talking about and the different pressure changes. All right. But I ran over a little bit, and we'll talk about that next lesson, as well as getting a couple of exam questions talking about what we've done today. All right. Right, perfect. So I'll just stop the recording now.